I would like to talk a little bit about integration, different types of data strategies in the context of cancer research. Before that, I would like to just clarify a bit of nomenclature that I'm going to be using. First of all, I'm going to be talking about different data modalities. And with this, I mean different data types, but I'm also meaning different ways of characterizing the features of a given tumor of a patient or characteristics of the clinical history of a patient. So for example, this could be DNA, RNA, image data, clinical data, anything. So I'm going to be talking each of these as a different data modality. And also I'm going to be talking a lot about signatures. And signatures in general, what I mean is a given biomarker, or it could be a function of several biomarkers, or it could be in general any characteristics that we think might be interesting to estimate, because it gives an important information about the status of a patient or the characteristics of a tumor. With that, I'm going to start talking about a little bit how do we use these signatures, this type of information in clinical research. So first, starting from the left, what we typically use are information signatures based on just one data modality. We have here on the left here the typical ER and HER2 for stratification of patients in the clinic, which is done with H&E staining. Pathologists just count the intensity and the number of cells that respond to the specific staining. And this is used to classify patients into ER positive and negative and HER2 positive and negative, and then to be allocated into treatments, hormone treatments, or targeted anti-HER2 treatments. This is just based on a very simple assay. Moving to more molecular subtyping, we have here the results of the first molecular stratification of breast cancer, the seminal study by Chuck Peru in 2000, where he defined the intrinsic subtypes, 5 subtypes. This was done just using the expression of a subset of genes obtained through a microarray. Basically, once you have this clustering, what they do is just they use correlation with one of the 5 centroids that they have defined, and so you can classify a tumor into one of these 5 intrinsic subtypes. And this is the basic of many genomic tests that you can see nowadays in the market, like Prosigna, Oncotype DX, or MammaPrint, which typically are just based on a small chip where you get the profiling of the expression of a number of genes, and then there is typically a logistic regression or something like that that will give you a probability, for example, of relapse. So one common characteristic of all these tests that I was mentioning is that they are based on just one data modality. And there are good reasons for doing that. It's because they are easy to implement in a clinical setting. However, us and others have seen the benefits of incorporating or integrating more than one data modality to improve the power of these estimates. For example, we have here the study where we just combined DNA and RNA, but more importantly, the correlation and the association between these two data types to identify a collection of oncogenes and tumor suppressors that will allow us to stratify patients based on those. And in this other study, we just used a multi-omic predictor just based on combining clinical data, DNA, RNA, and digital pathology to predict the response to treatment in the neoadjuvant setting. What we saw is that, of course, when you integrate more data type, these models perform much better. However, these are very difficult to be used in clinical setting because these, not only they are more expensive because they use more data, they can also be a logistical nightmare because you have to integrate many different data types and also different pipelines. So we have been trying in the past to see if we can go back to learning from integration of multi-omic data properties, interesting properties, and see if we can scale back those estimators into something that we can estimate using only one data type. So for example, here on the left, Cameron Young, who was an MPhil student, developed a classifier for the problem that I mentioned before, combining DNA and RNA, but using only one type of data. And Rong Zhu, who was a postdoc with me, so she worked on the problem of estimating homologous recombination deficiency in tumors using a series of models who go from integrating many data modalities and finding those biomarkers that are associated with the other data modalities to see how low we can go into a model which is simple enough and see if the performance is still good. So what we learned from these studies is that yes, you can scale down and go back again to just one data modality, but you lose some information there, of course. The performance is not the same. So what we like is to have a more comprehensive or systematic way of doing exactly this. integrating data, learning from signals of all these multiple layers of biological information, and at the same time automatically being able to estimate using only one data type. Because we think this is the perfect balance for performance and at the same time ability of translational into the clinic. So specifically, what we like is a general framework where we can identify specific signatures that are relevant to a specific task, using, if possible, just one data type. We want these signatures to be transportable or to be able to be generalizable to different contexts. The problem when you integrate multiple data types is that all of them they might have been carried using different technologies, and the more of these technologies that you put together and you use in your model, the more difficult it's going to be translated into different cohorts that might use different assays, etc. So we would like a model that will separate the task-specific signal from other signals that might be relevant to just that particular cohort or just that particular technique, for example. And how do we plan to do that? Well, the idea is to integrate auxiliary information. And we want this auxiliary information to be biological relevant and data-driven. That is, we don't want to be using general data sets like the pathway data sets, which might reflect general biology that might not be interesting for the task that we are using. We would like that to be more generalizable, of course, that it was interventional, causal, if possible. And very importantly, possibly, this auxiliary information might not be available for the individuals from whom we want to make the predictions. We want to stick to just one data modality. Okay, so with these requirements, Dom Kerkan, who is a postdoc now in our lab, but they did this when they were the PhD student, co-supervised with Sash Mukherjee. So Dom came up with a model that just filled all these requirements for the specific task of predicting drug responses on pan-cancer cell analysis. So this model uses the cancer dependency map. This is similar to what Manuela has been showing. So this is a very large database of many cancer cell lines from many different cancer types. And they have been profiled with many techniques. So you have all molecular data that you want, but we only want to use one of them, expression, okay? Because we want to make this as easier to translate into the clinic. So we want to use only RNA-seq data. So the task, as I'm saying, is so these cell lines are profiled using dose response drug curves for many hundreds of compounds. And we can summarize that with the area under the curve of IC50. So we want to predict exactly that, if a cell line is going to respond to a particular drug. The auxiliary data, or the one that we will integrate, is CRISPR screen data, which is exactly interventional data. It's causal. So this is for those of you who are not familiar with CRISPR, what it does is that knocks out a gene one at a time from the cell line. This is done in vitro. So we can measure if the cell is able to live without that gene. So it's basically measuring the dependency that that particular cancer cell line has on that particular gene. And this is very useful for identifying oncogenes, for example. So I'm going to talk about the models here. I'm going to give a very high-level concept. For specific mathematical details, I'm going to omit that. But yeah, I'm going to show you later some reference that you can follow. So the model is done in two stages. In the first model, so which Dom called a interpretable constraint variational autoencoder, what we have here is our RNA-seq data from all the cell lines. And then what we do here is we fit an encoder. Okay, so we come up with a latent representation. Now, the trick is that the latent representation is split in two parts. One is unsupervised and is going to reflect typical variability in the gene expression of all these cell lines. But then there is a part here that is informed by the CRISPR data. Okay, I will mention a little bit how that is done. So with this split representation, we can go back with the decoder to the original expression data. That is the first stage of the model. The second stage of the model, that I'm called prediction with interpretable constraints, what we have is a representation of the expression cell line is run through the encoder that we defined previously. And we use these latent variables that now some of them are unconstrained and some of them are constrained in form of the other data type. Then we can use a linear model to predict the drug response. Okay, that's what we do. So some technical details. This is a semi-supervised variational autoencoder, and the semi-supervised part comes from we are using a conditional prior on the CRISPR data. So basically, we have then a regression function where we are directly regressing the drug response, which genes with the variability in the CRISPR signal, how much a cell line is dependent of a given gene. That's how we do that. We use variational inference to train the model. And importantly, the model needs constraint selection. That is, we need to know in advance what CRISPR genes are going to be relevant to predicting the drug response on that particular drug. So the way that we do that is we do that through hypothesis testing. So we go and we compare, well, we take into account, we control for a lot of confounders that happen in these CRISPR experiments. And then we identify those genes which the addition of that particular gene from that cell line is predictive of a drug response. We are using that one gene at a time. This is just a simplification that we do because we want these constraints to be interpretable directly. So this constraint is going to be measuring the dependency that a particular cell line has on that particular gene. However, how this dependency is represented through the expression can be arbitrarily complex because that goes through the latent space. Okay, I'm going to show some performance of the model. So we see in two tasks that are not easy at all. So the first one is zero-shot learning of unseen cancer types. So don't design an experiment where we have at the beginning a series of cancer types, and we train the model on those cancer types. And then we try to predict drug response on unseen cancer types. And this is complicated because, as you know, some drugs are very specific on certain cancer types because there are specific mechanisms that are not amplified or are not mutated in all cancer types. So it's not easy. But this is a very good experiment to see if what we are capturing is biological properties, okay? Because if we can carry out through this constraint selection, if we can carry out biological properties. So here you have the performance of the model in a lot of different drugs. These are grouped by different target. And basically what we can see here is in general, the model does better than just using unsupervised with no constraints variational autoencoder or another baseline, which is a neural network. But what's important is that we can interpret, we obtain signatures that are relevant. So for example, here to predict lapatinib, we see that if the cancer cell line has addiction to ERBB2 or EGFR, which are the natural targets of this particular drug, we are predicting that these cells are going to respond to that particular drug. So basically what we see is that the unsupervised signatures are not as informative as the supervised signatures, as they should be. However, this doesn't work for all cancer types, and this is expected, because if the mechanism is not there for that cancer type, well, the information that we are carrying out with the constraint signatures is not useful anymore, and it could even give up to, well, predictions that do not work. So that's expected. And the second example that I would like is a task that, that we were mentioning before, that which is transfer learning on patients. Cancer cell lines are completely different to patients, but are these models learning some biology that can be useful to predict response on patients? And what we do here is the same. We fit the model on cancer cell lines, and then we try to predict in a clinical trial. This is the same clinical trial that I talked at the beginning of the talk. So this is comparing, we can compare here with our multi-omic predictor that is using a selection of curated signatures from the literature. What is interesting here is that here we are fitting the model using causal intervention on the cells. Here we cannot do these causal interventions of patients because we cannot do CRISPR on patients. CRISPR is done in vitro. We don't need to, because we are going to be inferring the dependency of the patients on each of the oncogenes that we know are important for prediction through the latent space. Okay? And what we see here is, first of all, you can see here that first, just checking here that the unsupervised part, this is a UMAP plot done with the unsupervised part of the latent space, and it's capturing biology, separating samples according to tumor type, to types of breast cancer, which is expected is very good. But then when we look at the performance and we compare with the multi-omic predictor here that using curated signatures, we see that the performance is very similar, which is remarkable because it means that we can identify automatically detect signatures that are going to be as good as the signatures that are curated by biology. And if you look at them, you see that there are some of them that are interesting, and we know that are predictive and make sense. Okay, I'm just going to stop there. And in summary, I'm just going to say that we present here a class of models, we could call it that way, that use auxiliary data to improve robustness and interpretability. Which is important, the auxiliary data, the data that we are integrating, we don't need it for prediction. It's only there for training and for learning the important biological properties that we need in order to make predictions. I presented here an application of the model for drug response, but the model is in fact very flexible. You don't need to use it with these data types. It's very arbitrary, so you can adapt it to different data modalities and different tasks. And we have seen, I hope that I showed you that the signatures that we generate are transportable across cancer, unseen cancer data, different unseen cancer types, but also from cell lines to patients, suggesting that probably the model is learning biological properties. Okay, so I'm going to just stop thanking everybody in my group, our funders, MRC and Cancer Research UK, and of course, Dom and Sash Mukherjee for this work. I'm happy to take any questions.